The overall goal of this procedure is to create biomembrane microarrays using a simple preparation method. This is accomplished by first coating silica beads with lipid bilayer membranes. The second step of the procedure is to deposit the lipid-coated beads on a silicon microwell substrate. The third step is to remove beads not in microwells using a polydimethylsiloxane squeegee. The final step is to image the lipid-coated bead arrays with fluorescence microscopy. Ultimately, this method can be used to determine binding constants for toxin-lipid interactions using an array imaging approach.
This method can be used to create arrays of spherical supported lipid bilayers and natural membrane particles derived from cells.
The main advantage of this technique over existing methods is that apart from fabrication of the microwells, the technique requires no chemical modification of either the substrate or the lipid membranes to create spatially defined biomembrane arrays.
The applications of this technique can be extended to label-free detection of lipid-protein interactions using surface plasmon resonance based on nanometric metallic hole arrays.
Though this method can provide insight into lipid-protein interactions, it can also be applied to other systems, such as studies of cellular focal adhesion.
Microwell arrays and squeegee preparations are detailed in the text protocol. This video begins with vesicle preparation. First, in a small glass vial, mix the component lipids for the vesicles. A total of 0.5 milligram of lipid is in this solution. Under vacuum, desiccate the mixture for 6 hours. Next, make a 0.1 molar sodium chloride solution and add 0.5 milliliter to the dried lipids. Allow the mixture to incubate overnight.